So I'm going to give a primer today on the basics of genetic variation and especially focusing on linkage disequilibrium and the history of the HapMap project. And if you don't know what the HapMap project is, hopefully you will by the end of this. This is, really now, work that took place, roughly 10 years ago. But it's foundational to everything that we do with genome-wide association studies and beyond today. Always important to start from the standpoint in which we entered into all this human genetics stuff, some of us several decades ago, which is that human genetics is, of course, not about prediction, even though that's often discussed. It's really about developing a much more fundamental knowledge base for therapy and prevention. And the way in which we look at this problem is that for many disorders, such as those of the brain, neuropsychiatric and neurodegenerative, for example, we've made very little progress in the development of therapies over the last 50 years, in point of fact. And that difficulty, and the difficulty of the pharmaceutical industry in getting new medicines onto the market, arises primarily because we don't fundamentally understand what genes to target. Now, when you read the newspapers, quite a bit more of the discussion is often surrounding personalized or individualized medicine that might emerge from the use of the genome. And while this is certainly a possibility in some cases, and I guess most notably in cancer, several dramatic examples of how this work have emerged, it's generally, for chronic disease, a secondary offshoot. And it's been vastly oversold to the public now for decades. But technological advances that you'll hear about in later primers are, of course, bringing this much closer to reality. So how does genetic variation matter? So why do we try to find genetic variants that confer risk to chronic diseases? Well, for a lot of reasons. One of which I'm sure you have heard about and you will hear more about, is that genetics, regardless of the strength of the effect of a particular locus on disease, polymorphisms in genes that confer risk or protection from disease link those genes and those diseases in a way that points out to us opportunities for rational targeting of therapies. Now, this doesn't happen overnight, of course, after we identify genes that confer risk to disease, but it does happen, and I think genetics has been proven over decades to be the primary source of insight into human disease that allows us to effectively target specific genes as having an influence on outcome. Genetics can really also be used to fundamentally, at a more basic level, alter the course of disease research. So age-related macular degeneration is a good example that I'll use for a few slides now. It's the leading cause of late adult onset blindness and will affect, in some degree, a very significant number of the members of the population, 5 to 10% to varying degrees. And starting with the first whole genome-wide association studies, which I'll get to in a bit, in 2005, common variation in 3 genes, complement factor H, complement factor B, and a gene of unknown function, were discovered very easily to explain half of the heritability of age-related macular degeneration. Now, what's very interesting is that the complement pathway is an extremely well-studied pathway in immunology, but had not previously really had any link to age-related macular degeneration, which, as I said, is a late adult onset form of blindness. But what's happened is that there's been a dramatic shift since the emergence of this genetic information. And now, for example, just to cite one example, instead of testing novel therapies for age-related macular degeneration on mice who have retinas that are burned out by lasers, now we can test therapies on a much more rationally based model that bears resemblance to the human disease, mice that are lacking the complement factor H gene. And in fact, those mice do display similar age-related retinal abnormalities. So this is a more fundamental way in which genetics can be used to create tools that allow us to fundamentally develop therapies. Finally, there are some examples where genetics can provide a personal risk assessment. the genetic associations in that complement factor H example were so profoundly strong that in fact we could identify some members of the population up here who had the combination of risk factors that placed them at 100-fold risk of developing disease compared to these other individuals down here. And that's, in the case of a common disorder such as AMD, a real and discernible value to individuals, because the individuals up here have close to a 50% lifetime risk, while the individuals down at this end have a less than 1% lifetime risk. And as prophylactic therapies, vitamins and so forth are developed and better understood, they can be targeted to those individuals. Now, genetics can also be used in other ways to understand positive and adverse responses to therapies, and also to predict individual dosages. Now, these types of tests are not often valuable clinically because we have other ways of monitoring, for example, the effective dosage in an individual of warfarin, which is a leading anticoagulant prescribed in the clinics. But the wide amount of inter-individual variation provides an opportunity for us to learn how genetic variation might influence outcomes in a different way. That being, can we predict the appropriate individual dosage? And in the case of warfarin, monitoring genetic variants at two genes, a cytochrome P450 that's involved in metabolizing warfarin, and the direct target of warfarin itself, the VKORC1 gene, an analysis can be performed, including other variables such as the individual's age, gender, and body mass, that actually predicts the effective dose in some individuals. And why this is important is that if you give an individual who needs a small dose a very large dose, you place them at risk of hyper anticoagulation, essentially risk of hemorrhaging. And if you give an individual who requires a high dose an intermediate dose, they don't effectively get the anticoagulating that they need. Yes?
Mark, you said that one use weight and body mass, and age and body mass. Do you know how much the genetic variation adds to the correlation beyond those?
Well, it adds statistically significantly. I don't have the percentage in mind because this is now a 10-year-old paper, but it was a pretty substantial enhancement of the ability to predict because those were in fact quite substantial. I can look it up for you afterwards, or you can read the paper. That was the point. The point of that particular paper was that the addition of the genetic variants was a substantial improvement to that traditional regression model. Now, it turns out not to be terribly practical because one can simply, as is done in the clinics, measure an individual's response week to warfarin. It's a bit time-consuming, but so is performing consenting an individual performing a genetic test, and ultimately, new drugs are going to come on the market, and they have already come on the market in this time frame that would require and have very different responses. So these genetic variants are not broadly applicable to anything but the response to warfarin necessarily. So those are some reasons why we try to find genes. But the question is, how do we find genes? And to give a historical perspective on this, we of course need to go all the way back to Mendel, who was the first to recognize that, in fact, there were some entities that were responsible for inheritance, and they were discrete, and he kind of conceived of them as grains of sand, but that they were very distinct and discrete units, such that traits such as the color or the shape of a seed or a pea were in fact transmissible in irregular, what we call now Mendelian fashion, dominant, recessive, what have you, and that the ratios of offspring were in fact very predictable from the phenotypes of the parents when the two organisms were crossed. Now, the first challenge to Mendel's rules occurred in something that essentially didn't make sense to Mendel at the time, which was while most of the traits that he was monitoring in the garden were passed along independently, and you could sort of imagine them as balls in an urn or grains of sand that you were randomly drawing to create offspring from a mating of two plants, there were certain pairs of phenotypes that appeared not to be passed on independently. And in the case of this particular cross that's exemplified here, you can imagine parents that carried colored and colorless seeds and smoothed and wrinkled seeds one might expect that one would get a quarter of each of those offspring if you were drawing balls from an urn, but in fact, almost all of the offspring, but not quite all of them, were either colored and smoothed or colorless and wrinkled. And why this was, of course, was that genes that confer risk to, or confer, these traits are, of course, not random balls drawn from an urn, but they're assembled along a chromosome. And the fundamental property that we still take advantage of in every genetic study that we perform of all flavors is the principle of linkage, and we'll expand on that in a bit. But essentially, genes that are on the same chromosome are passed along in tandem unless and until a meiotic crossing over event occurs in the transmission from parents to offspring of those chromosomes. So in this case, the genes of interest for the kernel color and the shrunken or smooth seed were in fact very closely localized on a particular chromosome, and that indicated that in almost every case, the parental chromosomes were going to be passed on undisturbed, except in the 3% chance that there would be a crossing over event in between those two sites. And so it took another half century before Morgan and his student Sturtevant, almost now just a shade over 100 years ago, published the first genetic map. And this was this is a historical paper in the field of statistical genetics and evolutionary biology, and that this was the first time in which traits, these being sex-linked traits on the Drosophila sex chromosomes, were arrayed in an order based on their inheritance and being passed on to a second generation of flies, were able to be arrayed in a linear order along a chromosome, and revealed that in fact the discrete units of inheritance of Mendel were passed on in a linear fashion on a physical entity that what we now refer to obviously as the chromosome. And so this fundamental first linkage map led to an entirely new and emerging field of genetics to really understand transmission on a physical basis as being entities along a chromosome, and leads to, inevitably, and now even decades and decades before we even understand the structure of DNA, a field known as family-based linkage analysis, which has to date been the major driver of our understanding of rare and inherited diseases with respect to chromosomal mapping. And so how family-based linkage analysis works is essentially you take a family, and hopefully, if you have access to them, multiple families with the same rare and severe phenotype, you identify the individuals that in that family, just within the family, that carry the disease or phenotype you're studying, and the individuals that don't. And the simple idea is that based on a simple Mendelian pattern of inheritance, dominant, recessive, or X-linked, what one can do is track down using DNA polymorphic markers scattered at fair distances throughout the genome. One can track down the arm of the chromosome and eventually zero in on the location of the gene that carries the mutation responsible for the disease. Now this strategy, first conceived of way back in the 1950s by Newton, Morton, and others who worked out the mathematics of how to take a family and a set of phenotypes and calculate LOD scores, was again decades in advance of any ability to molecularly track, to understand or discover DNA polymorphic markers and effectively screen the genome for a linkage. The first real successes happened right at the end of the 1980s with the localization and then eventual identification of genes for Huntington's disease, cystic fibrosis, Duchenne muscular dystrophy, and several others. And then through the 1990s, this strategy and follow-up molecular work to identify genes and mutations gave us in medical genetics a revolution of molecular understanding of the basis of rare and severe and inheritable genetic diseases. This all worked because the basic premise was that there was a single gene and that essentially the mutations were fully penetrant. And what that means is that there's a deterministic relationship between genotype and disease state. That is, if you have two defective copies of the cystic fibrosis transport gene on chromosome 7, you will get cystic fibrosis. If you don't have 2 defective copies of that gene, you will not, in any circumstances, get cystic fibrosis. So this means the mathematics works out in a very favorable way, and it's very easy to track the inheritance from generation to generation. And then because one presumes there's only one gene responsible, one can then amalgamate the information obtained in this way across multiple families. So those families may have different mutations, but those mutations are all pointing you to the same region of the genome and helping you track down the location. Unfortunately, despite a much greater effort in this particular line of thinking throughout the 1990s, whilst we were discovering the molecular underpinnings of literally thousands of rare and severe genetic disorders, we had essentially no success to speak of in human complex and chronic diseases that were more common and didn't adhere to Mendel's laws of inheritance. And this was quite vexing because we were putting a lot more effort into getting nothing out of these blue triangles than we were in getting things out of the pink squares. And even the simpler idea of let's find some mice that sort of manifest similar phenotypes and see if we can figure out their genes. That wasn't very successful either as we applied it to complex and chronic disease. And why this was, I think, is fairly obvious in retrospect. It's not that every disorder follows those rules of Mendel and is caused by a single mutation in a single gene. And if one simply considers that there might be hundreds of relevant genes for something as complicated as diabetes or asthma or of any type, and eventually even more complicated conditions of the brain, that individual genetic variants don't act in a vacuum, but they act sort of in concert with many other genes. And if you're looking at something like who had a heart attack at age 60, obviously a lot more goes into that question than simply what's contained in your genome. And one might need to measure intermediate phenotypes that are perturbed by the genome, but also perturbed by the environment in leading someone to end up in the clinic on their 60th birthday. So given that there's a much more challenging scenario that faced us coming out of the 1990s and our lack of success in understanding genes for complex traits, we as a field needed to go back and assess what do we actually need if we want to exhaustively study the genome and now look for, instead of perfectly segregating alleles in large families, look on a population-wide basis and understand where there might be subtle effects of genotype that, when they're amalgamated over the population, are very modest, but eventually read through and influence disease state in a not a deterministic fashion, but in a probabilistic fashion. And so, in order to affect this, there were several things that we clearly didn't have 15 years ago when we sat down to assess this. First of all, we were still in the process of sequencing the first human genome. So we didn't have any of the basic tools we have. And what we knew we needed was not simply a static human genome, but to understand how much DNA variation there was. Because DNA variation, however frequent it occurred, which we didn't understand at that time, was of course what conferred differences in phenotypes and disease risk from individual to individual. And it would be impossible for us to proceed down this research any further if we didn't understand how much variation there was and how to study it. Now, at the same time, we also didn't have any of the laboratory tools we needed to test DNA variation comprehensively. At this time, around the turn of the century, we were faced with laboratory tools that allowed us to maybe sequence or analyze a handful of DNA variants in a single gene. This was still a very expensive process and labor intensive, but it required that for us to produce human genetics results, we had to either guess the gene correctly in advance or rely and study and continue to study only those diseases that segregated perfectly according to Mendel's laws. And then, of course, we'd need to build a whole new suite of analytical tools and approaches that went beyond the traditional linkage analysis strategies of the past. So we started this process, as the last century closed and as the first human genome was being sequenced. And one of the first ways in which we started this was actually as a byproduct of the human genome sequencing. So it turns out the human genome, it was not one human genome or one magical chromosome that was being sequenced as part of the Human Genome Project. It was actually a library of a number of different individuals. And the way that you assemble a genome is to sequence short fragments of that genome, find where they overlap, and continue on and construct chromosomes and larger, what they called contigs. But those overlaps, when they occurred between chromosomes, two different chromosomes, they could be my two different chromosomes or chromosomes from two different people, different parts of the world, doesn't actually matter. They provided an opportunity for the first time in a widespread fashion to compare any two chromosomes at any location in the genome. And what was found was that there was actually quite a large amount of DNA variation. Once every 1,000 base pairs, when those two chromosomes came from different libraries, we were finding DNA variation. I'll explain what kinds of DNA variation. And the interesting thing was, it appeared that almost all of this DNA variation was quite widespread, in as much as it didn't matter where in the world the two chromosomes came from. And 9 out of 10 or 19 out of 20 of those differences that we would find in two chromosomes, we would then go out and rescreen a larger population, and we'd see that, in fact, both of those alleles, whether it was an A or a C at a certain position, were actually quite common. And this began to tell us that these were quite old DNA variants, not just new mutations that happened to have arisen in someone's recent genome. So what did the DNA variants that we started to find by this process look like? Well, the vast majority of them were just single nucleotide changes, which if you blink, you miss them. Essentially, here a T is changing to an A. That's what we call a single nucleotide variant or a single nucleotide polymorphism. Doesn't look so interesting necessarily, and 99% of these occur in intergenic sequence and probably have very little, if any, possible function. That wasn't the only type of DNA variant we find. There's obviously genetic variation comes in all sorts of flavors. Far and away the most common are those single nucleotide polymorphisms, or SNPs, but they're also probably far and away the most benign. And you can quite well imagine that a rare DNA variant that occurs once every 1,000 people, but which removes 500,000 bases from the genome, might be much more likely to have a significant function. And in fact, many of these other types of variants are much more likely to have substantial impact on the individual's fitness and underlie severe disease. Now, that sounds like a lot of variation, because if you say, all right, every few hundred bases we find a DNA variant, and you think there's 3 billion bases in the genome, that's tens of millions of DNA variants that possibly are conferring risks. So especially when you go back to where we were technologically 15 years ago, this was an impossibly large number to consider. When you put it in an evolutionary context, though, humans are just about the least diverse organism that walks the planet. And as you can see, if we were studying an experimental organism or a pathogen, or even our closest relatives, the chimpanzees, there's quite a bit more DNA variation in every other organism that you can imagine. And so while that's a lot of variation to study, it could have been a lot worse. And the other thing that we began to learn was that it was quite clear that, as we had suspected from those first results in lining up different sequences from different individuals in the Human Genome Project, almost all of the DNA variation that we were finding was quite common to all populations in the world, regardless of where they're geographically located now. And that sort of hinted very directly as empirical evidence of what everyone had long suspected, which was the out of Africa hypothesis of human variation. Now, of course, there's still quite a bit, also supporting that same hypothesis, quite a bit of DNA variation that is unique to the African continent. And this is, of course, because not everyone came out of Africa in the out of Africa hypothesis. Probably most of the sensible people stayed behind. And as a result, there's quite a bit more DNA variation in recently derived African populations than anywhere else in the rest of the world. And that's part and parcel consequence of the fact that in evolutionary terms, the human population has been very, very small for tens of thousands of years, hundreds of thousands of years, and only very recently, in an evolutionary sense, have we come out of Africa, spread all over the world, and had a magnificent population increase. Now, the other thing that we began to learn from those early studies was that, in fact, mutations, when we find them, are not only generally old, but they're also generally unique events, mutationally. And the way in which we see this is through an ability to track something called linkage disequilibrium, which I'll describe as sort of a byproduct of the linkage studies of an earlier era. And that is, when a mutation occurs, it occurs on a single chromosome in a single individual. It then gets passed on, and in some circumstances survives in enough offspring and their offspring and their offspring to become a more common DNA variant or polymorphism in the population. But if one zeroes in on the individuals that carry a particular mutation in the population, in almost every case, if one then compares the specific chromosomes surrounding those mutations in the people that carry the mutations, you see that those chromosomes are actually identical to each other. And I'll get into a little bit more detail of how this works. And that's again because of the same principles of linkage that I described, but now applied on a whole population. So nothing is different than what we described in the single family setting, except what one has to consider is that the entire human population is a very large and very deep family. So if we go back 10 generations, 20 generations, now there's been many, many more opportunities for meiotic recombination crossing over to create assortments of DNA variants. But this certainly still doesn't happen on a very rapid time scale because crossing over occurs only about once per chromosomal arm, or roughly once per 100 million bases in a meiosis. So if I say, two people are related by 10 generations apart, they're still, if they share identity at one particular point in the genome, likely to have on the order of 10 million bases surrounding that identical one to another. And so this is the property that's known as linkage disequilibrium. It's the same as the property of linkage, broken down by meiotic crossing over, but now any crossover that's happened any time in the chain, going back many generations, connecting those two individuals' chromosomes, creates a break in which now the two chromosomes don't match. But when you're very close to a mutation, you sort of identify all of the carriers in a population of a very specific mutational event, then when you compare the genomes of those individuals surrounding that event, they're all identical. And so, whereas when you draw two chromosomes, whether they're my two chromosomes or one of mine and one of anyone else's in the audience, and we find a DNA variant every few hundred to few thousand bases, if you were to take individuals that all shared the common mutation for cystic fibrosis, you would find that in fact those chromosomes right around the cystic fibrosis gene actually have identity over hundreds of thousands of DNA nucleotides, something that couldn't possibly happen by chance, and was turned into a very effective means of playing out the end game of the identification of mutations, such as the case of cystic fibrosis back in 1989. And that was, if most of the individuals who were unrelated to each other carried the same mutation, then by carrying the same mutation, that meant that they were actually simply related to each other at a more distant level, and one could actually figure out by surveying the chromosomes a little more carefully where those chromosomes matched, and that matching targeted the identification of the location of the gene very, very effectively. So while the linkage analysis initially would point everyone to a specific region of the genome, this final property of sort of applying the same strategy but to relatively distantly or unrelated individuals over many generations, linkage disequilibrium, was effectively used to track down a much smaller region of the chromosome and really pinpoint the genes and mutations much more accurately. So this all worked wonderfully, but everyone figured that it had very little relevance to any of the complex and chronic diseases that we were studying, which simply didn't provide us any signal whatsoever from our family-based linkage studies. So some of my interest in what I'll describe for you later on comes from research we started almost 20 years ago into the inflammatory bowel diseases, Crohn's disease and ulcerative colitis. These are chronic and highly heritable diseases of the gastrointestinal system. They're recurrent and incurable, but there are many different approaches to maintenance, but ultimately many patients, in the majority of cases, require surgery to either, in the case of ulcerative colitis, remove the colon, in the case of Crohn's disease, periodic surgery to clear blockages. And these are obviously very debilitating things when they happen. So we had, in one very unusual case, arrived at a region of chromosome 5 that we were studying based on a more traditional linkage study. And we had sort of selected to prioritize our studies of Crohn's disease because it was a relatively early onset and relatively high heritable disease that we thought was sort of a gateway into more complex and chronic diseases. And it turned out that turns out to be correct to a certain extent. And we zeroed in on a region that we had identified in early onset Crohn's disease patients as showing significant linkage, and began a process which was very laborious at the time, this is the late 1990s, in which we had to develop a sequence map of the genes of this region and then sequence the region in some number of individuals to capture DNA variants. So this was at a time the human genome was not yet finished. We didn't have a completely perfect layout of what all the genes were, and we certainly didn't have anything like dbSNP to go in and look at what all the DNA variants were. We still were at the point where we didn't have any idea of how much DNA variation there was in the human genome. And so we had to perform essentially manual sequencing in short segments across this region to identify polymorphisms and to look at their patterns and to see if any of the polymorphisms, rare or common, might describe and explain the association that we found. What we found was actually quite a bit more interesting than just that. When we began to line up the chromosomes, and this was a population of unrelated individuals from the metropolitan Toronto area. So this was just about the opposite of an isolated special population such as the ones that I was describing earlier, and there was no reason to think that these individuals would share any of their genome more than chance, because we started by simply looking at population control chromosomes before we even got to the disease cases. But we found some very interesting patterns of haplotype diversity, and I'll walk you through what I mean by that in just a second, in which there were certain regions where if you slide a window across, you would find that there were a very large number of diverse haplotypes, and then other regions, when we slid the window across, where there were actually very, very few distinct chromosomes in the population. And so one example of those was a segment of about 80,000 bases, where we would expect a huge amount of variation and so forth. And we found that when we kept adding additional polymorphisms to this particular segment of the genome, almost everybody in this completely outbred and heterogeneous population had one of two very distinct sequence patterns across the region. And I'm displaying here only the bases that vary in the segment that we were testing. There's obviously a lot of bases that are not variable. As I said, only 1 in 1,000, 1 in several hundred bases change. So for the purposes of these pictures and many that you see in the literature, we often just draw pictures of sequence, assuming the 99 point some odd % of bases that aren't variable from person to person. So the fascinating thing was that over the time frame of human history, there should have been recombination happening throughout this region, peppering the region, and creating all sorts of new combinations of haplotypes than we were actually observing in reality. And so something profoundly different than the expected model of human recombinational history was going on. And then as we extended those regions, we did ultimately come across short intervals in which it was apparent that almost all of the meiotic crossing over events were occurring, because we could see quite naturally then a continuation of patterns in many individuals that were essentially reversed in other individuals. And so this gave us some fundamental insight and began a process by which we calculated haplotype maps, and this led to the haplotype map project, which I'll describe in just a moment. And essentially, what we were finding was that if we sequenced a region and then explored the DNA variants in a very large population of individuals, and again, this being a completely outbred population, we would find that it was the rule rather than the exception that we could partition this segment of genome, now being almost 500,000 bases, into some very discrete segments where there was very limited sequence diversity. And so these were telling us of segments that in some cases contained multiple genes, where recombination simply was not active very often, if at all, in the human population. And then interspersed were short intervals where almost all of the recombination was happening. And it turned out that while this was objectionable to mathematical modelers as a sort of an anti-conservative model of how recombination would occur, it was quite standardly understood by anyone who had tried to perform fine recombination mapping in mouse crosses or in yeast crosses, that this was always the way in which recombination worked. There were specific hot spots and there were long cold spots in which recombination was very inactive. So we pursued this down to identification of one of the first common risk factors for Crohn's disease. The other side of this nice patterns of linkage disequilibrium is that you can't really distinguish one of these DNA variants from many of the others. So we didn't know which of the specific nucleotides was responsible for conferring risk to Crohn's disease, but surely one of them was, and this ends up at this very early time in one of the most replicable loci in today's Crohn's disease studies, to which we've now vastly exceeded 100 similar associations throughout the genome. But more importantly, it led us to focus, David Altshuler, myself, Stacy Gabriel, and many others here, on a project that came to be known as the HapMap project, which was basically, if we could effectively study the genome and partition it in this way, it would then be the case that we might be able to perform association studies very inexpensively by simply intelligently surveying a few variants from this interval, a few variants from this interval, and so forth. And in a much more affordable way, study the vast majority of common DNA variation as it pertains to various complex diseases. So the HapMap project emerged around 2001, 2002, as that first human genome was being completed, as a follow-on project that was done exactly in the same spirit as the Human Genome Sequencing Project, and to develop basically a freely available public resource of DNA variation. And so the initial goals of the projects were to perform a high-density map of single nucleotide polymorphisms, because they were easy to identify and assay in the lab, that would provide information about which of those variants were valid, what their frequency was in different populations in the world, and create a resource that would be used by medical geneticists everywhere to study complex disease very effectively. And the important thing, which we focused on throughout, was that this was really meant to be a resource that would help accelerate the genetic discoveries in complex diseases. There was no specific goals unto themselves, other than those which would accelerate that end process, not which were internal to the project itself. So that in that way, it was somewhat different than the Human Genome Project, but very much in the same spirit. So in order to effect that, of course, we were among the early adopters of what are now standard practices in the field of making all the data essentially in real time available through project websites. We developed and distributed tools here that made that data more usable and effective in individual labs. And we did quickly come to certain observations, such as those patterns that we initially described anecdotally in that region of chromosome 5 about recombination hotspots were actually the rule throughout the genome and not simply an exceptional feature of that one region of the genome. So now we transition to the second half of the talk, which is a historical primer on the HapMap. And so when I used to give this set of lectures, it occurred in multiple lectures because, of course, in the latter part of the last decade, using the HapMap was kind of an everyday thing. So now I've shifted more to doing this from a historical perspective and showing you how the fundamental data and insights that we achieved as part of the HapMap project have been used to construct the way in which we go about human genome association studies, in particular genome-wide association studies. But the day-to-day utility of the data is not something that most of us, even including myself, deal with any longer. So the HapMap project, from the outset, drew on some original, some longer-term observations in population genetics that human populations are extraordinarily diverse, and you can sample from all over the planet, and you would find exceptional diversity and a lot of interesting things. But that if you needed to pick a small number of individuals and you wanted to get as much diversity as possible, the sensible thing to do would be to sample some individuals from Africa, some individuals from Europe, and some individuals from East Asia, because population migrations have gone their separate ways for thousands and even in some cases tens of thousands of years. And so in order to do a relatively small project, which was all that technically we could do at the time, but to have it broadly useful, these were the populations that were chosen. And a historical timeline, just to give you a little construction of how this project took place, was that primarily it took place almost completely contained in the last decade. Almost all of the critical work took place in the earlier half of the last decade, but the project continued on to expand the resources. But by this time, the technology had really developed in a much more effective way, such that the effort was considerably more to get us to this first phase map than it was with anything that came afterwards. So if one is building a human genome sequence, one knows one is done when one has a complete sequence of every chromosome or something reasonably approximating that, and we knew how big the chromosomes were. When we were set about to build a variation resource, there was no such guarantee that we understood when a variation resource would be complete enough. We'd want it to be the case that the resource we were creating described most DNA variation, at least most common DNA variation in the human genome, but there was no one to tell us how much that was. So what we needed in parallel with surveying the whole genome was a very detailed sequence-based look at some representative number of genomes. And now, while it's easy to imagine sequencing a genome over and over again, bear in mind we had just come through 10 years to sequence one genome. And the idea that technologically we could sequence, even 100 genomes was still prohibitively expensive and much more than the budget of the entire HapMap project. So what we did was we picked some typical regions, and this is actually a precursor to what's still called the ENCODE project of the day, which was a project to apply every technology available to understand genome variation and then eventually other types of DNA-based variation to a small number of representative regions that span different chromosomes, different rates of recombination, GC content, and other things we thought might be relevant. And so with this sequence-based data, as we customarily still do today with emerging whole genome sequence data, we assessed its completeness by seeing how much of the variation that was going to be in our resource and was in the nascent dbSNP database at that time was captured if we focused on variation seen in a single individual, variation seen in a small percentage of individuals, less than 5%, 5 to 10%, and so forth. And what we found was that happily at the time, it wasn't very hard, and in fact in the database directly were the vast majority of common DNA variants, but very clearly, and still is the case to this date, the majority of rare DNA variants are neither directly themselves in the public databases, nor do they have a correlated SNP. And the idea of a correlated SNP is a very important one that's fundamental to everything else that's going to come in the talk. And that is that, I'll come to it shortly. But anyway, as with those patterns of variation in chromosome 5 that I showed earlier, there was a great deal of redundancy among the SNPs, that if you type this one, it sort of predicted what was going to happen at the next one and the next one further down the chromosome. So you didn't, in fact, necessarily need to examine directly every single SNP, but if you examined something that was highly correlated to that SNP, you would get the same value in an association study. And again, as I said, we focused on single nucleotide polymorphisms, but we're creating a resource by which we could use those to essentially flag patterns that represented insertions and deletions that were small or even larger in some cases, and other types of repetitive DNA elements, all of which tracked with the same human history of recombination. So The purpose of this was to accelerate those genetic association studies to disease. And the importance of the patterns that we were seeing in an association study can be succinctly summarized in this way. If we're performing an association study and there's a causal variant hiding somewhere in the region of the genome that we're looking at, we will find that association, that difference between cases and controls, if we examine the causal variant itself, or if we examine another variant or haplotype that's close by and highly correlated to the variant that's actually causal in that region. Because if two variants are essentially identical to each other in terms of their DNA variation pattern, if one of them causes a disease, the other one is going to tag along and show the same difference between cases and controls in its frequency. And so we use the traditional correlation coefficient, or R squared, to tell us how closely related two polymorphisms are. And that if we were to build the haplotype map that we were proposing, we wanted it to be relatively complete. And we assessed that completion rate by the proportion of variants that would be highly correlated to a hidden variant from that sequencing exercise. So basically by taking some variants out and hiding them, we'd ask, if we built a HapMap, how often would we have a highly correlated variant? And what would the average correlation coefficient for a randomly hidden variant be? As you can see, these were very high numbers, which meant that we would have a relatively thorough ability to perform association studies if we built the resource we wanted to build. So in applying the HapMap, then, we worked to expand on its use in a number of different ways that I'll just go through relatively briefly, because some of them we use today, many of them we don't, but they provided a foundation by which we perform almost everything that we do in genome-wide association studies today. So the concept of tagging, then, was a natural byproduct of these correlational and linkage disequilibrium relationship. And essentially, it's better to sort of pictorially show you how this works. And that is that if we survey a region with 6 single nucleotide polymorphisms closely spaced along a chromosome, and then lay out the 4 different haplotypes, or the 4 different variable sequences that we see in the population, what one sees is that if one has an A at this location, in every case, that chromosome continues on and has a G in this location. And if one has a T in this location, that chromosome continues on, and there's an A in that location. That's a very high correlation between those two sites. And what it means is that essentially, if one is performing an association study, one doesn't actually need to look at these sites if one looks at these sites. Because if I have a reference panel from the HapMap that says, I know if there's an A here, there's a G here, one can simply computationally fill in the G or A at this location after one examines that particular site. And so that actually creates some considerable efficiency in how we might construct a genome-wide association study, but there was much more to that efficiency than necessarily met the eye. So if one now has eliminated the truly redundant SNPs, where there's a one-to-one relationship between this SNP and this SNP, this SNP and this SNP, and this SNP and this SNP, one can then find further relationships in the data, such as only the chromosomes that carry an A and a G at these 2 sites go on to carry an A. All other chromosomes go on to carry a C at that 3rd site. So in point of fact, if we know that piece of information, we can then ignore the genotyping of this polymorphism as well and fill that in computationally. And so this created opportunities for much greater efficiency, because now you could take whole segments of the genome, tens or hundreds of kilobases, and capture all of the genetic information in a simple pattern of 2 or 3 single nucleotide polymorphisms systematically. And this allowed Paul de Backer, who is one of the leading postdocs working with David and myself on this project, to calculate just how much effort it would take to perform a reasonably thorough genome-wide association study. And you could actually do a creditable job with 300,000 well-chosen SNPs. And it was certainly no accident that the first substantial genome-wide association array from Illumina put out around 2005, 2006 had 317,000 SNPs on it, well chosen in the way that Paul in this paper had recommended. Now that would only be a useful tool if the relationships that we use to pick those SNPs were consistent across populations. So we selected some group of European people that were living in Utah to represent all European people dispersed throughout the world. That might be a very ill-advised thing to do if those patterns didn't maintain themselves. And so at the same time Paul was performing that study, he was also asking this question of whether or not those relationships, if two SNPs were highly correlated in the Utah individuals, would they be correlated in other people from Utah? They very much were. Would they be correlated in self-described whites from Los Angeles? You can imagine that might be a slightly more diverse group than the individuals in Utah. But in fact, those same relationships were maintained. And in an isolated Swedish-speaking part of Finland, the same relationships were maintained as well. So whether you went to a more diverse or a less diverse European population, we could identify that the same patterns were there. And so these were really fundamental insights that led to how we designed the first and how we continue to design genome-wide association study arrays. And that was, in fact, the pressing question as the HapMap drew to a close, was would there be technologies? We could pick the SNPs, but would there be technologies that would allow us to perform a whole genome association studies? And I think I can very quickly go through the fact that starting around 2005, there were in fact very effective technologies developed by Affymetrix, Illumina, and other companies that allowed us to conceive of, for the first time, putting a drop of blood or extracted DNA from a drop of blood on a single chip, as in the case of that Affymetrix chip, and actually assessing hundreds of thousands and now even millions of DNA variants throughout the genome in a single experiment. So just an incomprehensible amount of data compared to where we had been just a few years before. So the technology came along, that was great. We could inform from the data of the HapMap project how well to design those studies, but we could also then use them on the backside in a different way to assess the coverage of those studies. And essentially, if we were to take the SNPs that the companies happen to put on those products, then genotype the same samples as in our HapMap resource, we could then use the other SNPs in our HapMap resource that were not on the array as sort of an unbiased estimator of how well covered the genome was by those particular products. And we went through the same analytic process as we did in the development of the tagging to assess basically indirectly by single variant correlation and then more completely by those multi-variable combinations, how well one could perform in a genome-wide association study. And while we found that we could do a reasonable job just with those single marker relationships, it was quite clear that we needed to go beyond those and look at those tests of haplotypes, or those predictors of where multiple DNA variants in a region were predicting the outcome at other DNA variants that weren't themselves directly one-to-one correlated with one of the single nucleotide variants on the arrays. And when we did that, we certainly could see that we could substantially improve the rate at which we predicted other variation not on directly assessed on those genome-wide typing arrays very substantially and get to the point that Paul predicted us to get to, which was a reasonably complete genome-wide association study covering 80+% of genome variation, of common genome variation, with a single experimental strategy. And so these types of analyses, these relationships of individual variants and then multiple variants to other variants that you didn't examine, these were the forerunners essentially of what's widely used today as imputation technologies. And imputation technologies are simply more mathematically advanced and sophisticated ways in which reference databases of complete sequence are then examined with respect to new individuals who might be genotyped only at a small number of sites. And essentially the results at DNA variants in those new individuals that don't have complete sequence are extrapolated from the reference database and from the genotyping data that those individuals do have. Now, there were many other applications of the HapMap data and the relationships between variants to variants throughout the genome. The most obvious was that as we started to get the first results from those DNA typing experiments performed with genome-wide arrays, we were looking at still only a few 100,000 variants, and it was critically important that we understood for every single nucleotide variant in that association study, what that variant represented. Essentially, what was it describing to us in terms of variation and other, nearby neighboring polymorphisms, whether the marker and this particular part of the chip was tagging or closely linked to particularly important coding variants or other functional variants nearby. And we explore a number of techniques, some of which are still used, to improve our understanding and improve our ability to perform association studies by understanding how much DNA variation and what DNA variants specifically are tagged or correlated to each SNP that we genotype on those arrays. Now, a more important and fundamental thing that I'll come back to and demonstrate in a moment is performing an integration of two different association studies results. Now, in the early days of this project, and I'll give you an example of this, it was critically important for us to harmonize reports of association to single gene studies where individuals had, for a time, taken 5 or 10 randomly selected polymorphic markers in and around a gene, genotyped them, and made a claim about association to a disease. Others would come along and try to do the same experiment, but they would pick different polymorphic markers. And without a reference with which to connect those polymorphic markers, we had no way of knowing how to look at these two results side by side. And in fact, the HapMap and the reagents from the 1,000 Genomes project that have come since give us the substrate. This is very sort of analogous to the way in which we think about the imputation technologies, give us the substrate to understand the relationship between reports of association. So if I were to, for example, describe that I saw a compelling association to the 4th single nucleotide polymorphism in one of those red ones, and you were to then publish a paper that you saw an association to a different variant but one of those red ones, single nucleotide polymorphism, the reference database would tell us that evolutionarily and in a real sense on the sequences of the individuals, those two variants are essentially mathematically equivalent to each other. So our observations of association would have been concordant and consistent. Whereas if after my initial report of association to one of those red-colored variants up there, someone else said, oh, I tested variants in that gene too, and I saw association to, this variant over here, then one could imagine that there was a time without a reference database when people would say, wow, those DNA variants are very close to each other. That must mean something very important is going on with respect to disease there. But in point of fact, the observations are actually in stark conflict with each other because my associated variant and your associated variant were completely different. And unfortunately, this was the state of play in human genetics without a reference database. And so in the early part of the last decade, there were many noteworthy examples, and I'm just going to give you one to sort of articulate just how important it was that this type of data became available in a very public and widely accessible fashion. So there were a number of studies in 2003 and 2004 that came out describing an association of this gene, which is conveniently known as dysbindin, as being associated to schizophrenia. And obviously, a molecular link to schizophrenia was something that we did not have, and which is a critically important target of molecular genetics, because schizophrenia falls into one of those categories of diseases that we don't understand, that we hadn't, up until very recently, understood any of the molecular underpinnings of, and therefore had failed dramatically to develop therapies. So there was extensive linkage to disequilibrium across this dysbindin gene that described in the HapMap project, and patterns of DNA variation that we could easily see once we had created the HapMap fell into patterns similar to that region of chromosome 5. And across the entire gene, the human population could be essentially partitioned, if you took all their chromosomes and lined them up one after the other, into a few different bins that chromosomes, the majority of chromosomes either had this pattern or this pattern, which were related to each other but different by a number of steps evolutionarily, and smaller fractions of the population had these other distinct variation patterns that were more evolutionarily diverged by mutation. So the first association came out, and there wasn't the benefit of this, HapMap reference. So there was no way in which the authors could have imagined what was to come, and they didn't like it very much when it did eventually come. But the initial reports of association were to random variants in and around this gene dysbindin, and this was very widely covered and very excitedly, recognized by the psychiatric genetics community. And then, so because this was such a landmark potential finding, many other groups began to study the association of dysbindin and schizophrenia, and hopefully their computers work better than that. All right, we'll get back to that. And when these papers were published, they described different sets of variants being tested and associated to schizophrenia risk. And they were widely received as replications of the original finding. But in fact, this paper, which was a very oft-cited replication of the original paper, was actually describing association to schizophrenia of very different DNA variants. They were in fact unrelated and actually, sort of would, if you put them together, wash out the signal for association. So many other groups started to get excited when this paper came out and started performing their own association studies. And they also described positive associations to dysbindin, but to very different patterns of DNA variation throughout the gene, none of which were consistent one to another. And if you added them all up, you would get no signal whatsoever. And then ultimately, a paper was published that was still described as replicative, but actually saw the exact opposite effect, that every other variants, except those originally reported, were associated positively to schizophrenia. So these were entirely inconsistent findings, which once we had the HapMap, we were enabled to pull together and ask the question in a harmonious fashion, is there any evidence of a consistent nature to dysbindin risk? And in fact, the answer, as you can pretty well imagine from this cacophony of discordant inputs, was that there's actually absolutely no evidence of association to dysbindin, despite 5 to 10 papers published claiming a positive association. And so while we often dwell on the successes of finding genes that have come since the HapMap with the foundation of genome-wide association studies and the explosion of findings in individual studies, here shown type 2 diabetes and Crohn's disease, the further explosion of meta-analysis of genome-wide associations, all of which you'll hear about in upcoming weeks, we can't overlook also the fact that having these reference tools and databases has been integral in cleaning up a rather desultory history of human genetics claims and really providing an unequivocal way in which to compare the results from one study to another and allow the types of techniques which we consider routine today, that if you perform a genetic study and I perform a genetic study, regardless of whether we typed the same DNA polymorphisms, we're able to put those studies together harmoniously through the HapMap database and through now the 1,000 Genomes database as well. And so the challenge we set to ourselves 15 years ago was, of course, we needed to understand the genetics of complex and chronic disease in the way that we had begun to understand it in severe Mendelian disease. And happily, as we could see from the early trajectories and returns in 2007, and then obviously what's transpired over the last 5 to 8 years since then, there has been dramatic progress in the identification of genes and ideas, concepts, and hypotheses for how therapies might emerge from that genuine molecular understanding of disease. And so with my thanks for many of my colleagues and trainees at the time of the HapMap project, earlier and slightly thereafter, who made instrumental and seminal contributions to this area. I would be glad to take a question or two before we transition the morning.